Okay, good afternoon, everyone. We're really happy to have you along with us today for our final Science and Innovation Center webinar. So we were hoping to get some of this out in Philadelphia earlier this year, but of course with the COVID, we've moved everything to become virtual. And so this is sort of the last of several SIC's webinars that we've put out. So I'm really excited to have Blanca Camoretti-Mercado at University of South Florida and Jason Bates at the University of Vermont today. And they will be talking about artificial intelligence and where things have changed over the past few years and where things are kind of going in healthcare and in research. So without further ado, I'm going to pass this over to Blanca for the first part of the presentation.
Thank you, Mike. And thank you, Liz. If you can project my slide, I would appreciate it. And welcome everybody. And thank you for joining us in this session. So can you go to? So I would like to do first an introduction, broad introduction about artificial intelligence, and focus a little more on the healthcare component. And I think Jason is going to go more into the application in biomedical research and in the imaging, in which area artificial intelligence has found a very fruitful area to give contributions. So may I have the first slide, please? So we all know that we are living in this new revolution of the digital era. So we go back 2 centuries ago. The first revolution was in the 19th century. We have the Industrial Revolution, in which it was characterized by the advancement of the machines, which allowed mass production of goods which could reach the hands of people. This was in our medical field and research. It could bring, for example, better surgical tools and the evolution, the development of the germ theory of disease. But as any advancement, it has also some drawbacks. And we could probably state at this time the beginning of the pollution that we are battling today. And this progress brought up to cities more crowded, so it makes spread of disease much easier. In the 20th century, what we have is the area of the information, in which these machines become more tiny, more efficient, and we're able to gather and store information in large amounts, not only about ourselves, but also about the environment. This, for example, is the development of the television, the cell phones, etc. And in the medical field, we have the beginning of the electrical medical records, and for example, the advent of the antibiotics. On the risky phase of this progress, we can see that this advent of massive information couldn't be handled with the same capacity and speed, and the process of the data was also falling behind. And somehow we saw a decrease in time in the interaction between the patient and the physician, because they have to be more taking care of the digital data that is coming through the medical records. And also we saw some kind of unethical use or execution of human studies. In the current century we are right now, the digital or the data era is characterized basically by this avalanche of new information, of new data that is faster, bigger, and allows, yes, more communication. And with the facilitation into integration of those data, now we have the appearance of the electronic health recordings. People can learn about themselves, about medicine, about disease online. But this, the drawback of all this is that people are, having more sedentary lifestyles. We are start dealing with privacy issues, and of course all the misinformation that occurs, and all brings somehow to the dishumanization of the individual. The next slide it shows then the type of data that we are talking about. There are basically two types of data. The structured data are the ones that we are all familiar as a physician, as a scientist, and that's the data that we can put very easily in tables. It's basically the one that we have rows, we have with the different observation, we have columns with the different attributes, the different features, different characteristics, different variables of that data that we are collecting. The unstructured data is the data that cannot be put in tables in the tabulated form, and actually becomes the data that is the more abundant this day. Is, for example, the Facebook, the imaging, genetic data, notes from the doctors, EKG signals, etc., etc. Is the data that is completely free, disorganized, and as you can imagine, it's very difficult to handle this data because there is no an organization and there is no a hierarchy. So how can we manage this data? In the next part, we can see now, Elise, please. We can see that via a feature extraction, we can transform the unstructured data into structured data. Here, for example, you can see there a paragraph with 5-7 sentences. And how can we make this test into which completely unstructured? There is information there hidden. So how can we structure that information? For example, what we can do is to do a table with word counts. So we can make the counting of each or some of the words, see how many times they show up in that paragraph. We can maybe calculate the average length of the word in each sentence. We can probably maybe add a new feature like a topic. And if you read this one, it's about astronomy. It's talking about the morning, about the stars, Venus and Saturn, etc. So there is some ways that we could transform unstructured data into structured data because we know how to deal with the structured data. Next one. So the structured data is the one then that we are going to handle. And there are basically of two flavors. There is the qualitative and the quantitative. The quantitative, of course, are the numbers, which can be discrete or continuous. Examples of that are the temperature, the score, the age. Is easy to distinguish because we can do math with them. And then they need, by their nature, a little processing. So instead, the qualitative data that are structured are also very useful. And they are usually not numbers. Also, it can be numbers, for example, a zip code. But the difference is that making math operation on them makes no sense. Adding, 2 zip codes makes no sense. Adding 2 telephone numbers makes no sense. Multiplying that makes no sense. In the quality data, for example, we have categories. It's categorical data. It's nominal data. It's, for example, sex, color, that type of data. And they in order to do parametric operation with them, we need to process this data. Next slide, please. So as important as the data is the metadata. And the metadata, simply said, is the data about the data. It's, for example, what is the data is about, who created, which purpose, what type of data is there, which are the columns, which are the different observations, how many of them are there, when was modified, where was archived, who did it, who used this data, how many queries were done about this data in the last day, in the last month, etc. What is this data? This data set, is there related to another data set in a particular data bank? how large is the data set, which is the primary key, which is the most important column feature of that data. And this is very important because it tells us information about the data itself. And it's very important if we wanted to go back and, for example, repeat some information that was obtained about the data, some results that was obtained about that data. So, and it can be, data, it could be statistical, result, could be structural, could be administrative, names, people, could be reference, could be descriptive, etc. So next one, please. So, and why is the data important? I don't know if you are familiar with this pyramid of knowledge. At the base of the pyramid, we have the data, which is, in our case, could be, for example, data coming from the different omics, could be data coming from the electronic health records, could be the Internet of Things, could be all the data that's coming from the mobile technology, from the cell phones, from the watch, the Apple Watch, etc., the sensor data that we have available. This data, as such, is just observations, are events, and there are information hidden in there, but only we can get that information if we interpret that data, if we make context interpretation of that data. Now, once we have the information, which is a big step from the data, that is not enough because we need to organize that data, to synthesize, to process that data, and that gives us the knowledge that we could use. So many people stop here in the knowledge, but for us, scientists and physicians, we know that we need to go one step further, and that is to use our judgment, to do our knowledge, to bring wisdom to how we use that data, how we use that knowledge, how we use that information. Up to the knowledge is where the data science is helping us, but it's the human component, the judgment that comes from the individual, which make this pyramid, I think, a more complete. And in the next part is that what kind of how can we could organize this knowledge. Next slide, please. So, and this is, for example, the classifiers. The classifiers are how we could organize this knowledge. And I, put some of them up there that I will try to get into them. We're all familiar with regression, which could be linear regression or logistic regression. We have some knowledge of clustering, how data can group together based on a particular characteristic that they share. The decision trees are very nice models that are very intuitive and very easy to interpret. And then we have the artificial neural networks, which Jason is going to speak a little in more detail next. Next, please. So definition is important because artificial intelligence is a kind of early discipline, and there is a lot of confusion still depending which book you read, with talk you listen, etc. So data, as I say, is the observation, is the collection of observation that could be organized or disorganized. Artificial intelligence is a field that actually, as his name indicates, try to create, to use, to recreate the abilities of the human brain applying to the data. Machine learning is a collection of methods that try to give the ability of computers to machines to learn from the data without necessarily the programs of the people giving information to the machine. And the way that the machine does is by data mining, is by going inside the data and trying to find the relationships, the patterns that are hidden in that data. Cloud computing now becomes much more popular and accessible because of all these amount of the data, the storing, the access of the data, and the program, the software, can be done via the internet. Because sometimes having our own personal computer, laptop, is just not enough. We need much more power and capacity. And the big data actually is referred to the data that is too large to be processed by a single machine. If you have a data set and you want to do some kind of work with that data set, and suddenly your machine crashes, even though with all the memory and the power you have there, probably you're dealing with big data that is characterized by these 3 V's. The volume, we are talking about, gigabytes, terabyte, petabyte of information. It's variety, meaning that we have data of different format, images, sound, numerical data, et cetera, et cetera. The velocity is that this data is coming at fast speed. Every millisecond, data is being generated in large amounts. And now also there are more V's being added to these 3 classical V's. The veracity, which means how useful, how truthful, how faithful the data is, because of this, velocity, especially that could impinge some, vulnerability to the data. And the visualization will be becoming more important because it's hard for us humans to try to visualize the information that big data is giving us. So you will see that a lot of ways to visualize, a method to visualize the data. Next one, please. So in biomedicine, this big data basically falls into 2 different buckets. One is all the omics, which is, the genomics, the epigenomics, the transcriptomics, the lipidomics, metabolomics. That we can also add the microbiome and the phenomics. And then next one, please. We have also the electronic health data, health records, in which we can collect the physician notes, the information about laboratory data, about insurance, imaging, vital signs, etc., etc. Next one, please. So about the clinical big data, the advent of the EHR, EWatch would make this possible. And I think that the potential and the benefits of this, utilizing this opportunity is just, endless. That is because the electronic health record are used basically universally in all the developed world to capture information about patients. And it's relatively very, very cheap to do that. And there is, and the data is collected longitudinally and for a long time. And it's all type of information that are contained in this type of records. And the records are very reliable. And also, what is very useful, they're standardized, which means that we have an accurate phenotype, accurate diagnosis. But it's not only medical information about the patient that is collected, but we also have, for example, other type of data about insurance, about billings, about auditing, which makes this opportunity much larger than only medical data. And this brings the opportunity to do studies that are, for example, independent of the disease, because the electronic data is, disease agnostic, could be applied to any data, so any disease. So we can study virtually any disease. And because of the volume of the data, it could be the opportunity to do population studies are basically, very possible. So the translation of study from the medical to the population, the T4 step of the translational research, is really an opportunity. Next one, please. So, but this capability of doing this large scope studies with medical information, it kind of reminds me what you know, in the business area, the technique area, you know, the big company like Amazon, Google, and Netflix already have done. And it's very nice exercise to try to see what would be the opportunities for healthcare system and what we can learn from these other, you know, business enterprises. And I think that one of the important difference is that for example, in Amazon, the data is real time, is online, and it's coming from searching and purchasing from the user, the customers. While in healthcare system, it's a completely different you know, world. These are private information, medical information that is not simple. It's not just a simple click. It's not simple score that is a low density, but in contrary, it's very rich information and requires, most of them, because an instructor requires processing. In the business world, we have the customer is willing, giving the information, is free, is open access, and he or she gets benefit because all that information is centralized in a particular vendor, and he receive whatever they want in terms of personalized outcomes, recommendations, etc. That is very different in the medical field, in which to obtain the medical information is very costly. It's not cheap. The personal data is private. It has to be protected. Usually, this is not centralized. Patients receive care from different providers. So the data are in different hospitals, for example, different institutions. And yes, the benefit could be really also personalized, the same as is in the business. Next one. So then here I try to list the different challenges that big data in medicine, in biomedicine, faces. One about the capture and the collection. Cleaning the data is very important. The data is not complete. Sometimes is erroneous. It has to be clean. And usually 80% of the work that is done in time and in effort in preparing a data set is just cleaning the data. The other 20% is about the analysis, getting the result during the queries, etc. And there are many factors there that are not too much to our concern, physicians and researchers, but more, the IT department, about the storage, the sharing, the updating, etc. But for us, the most important is about the security, and this is true among any type of big data. The security of the data that is, stored. Next one, please. So, and in the last 5 minutes, I want just to do very quickly about some of the, what this big data can do that for us. One of the big things is classification. The other thing is forecasting, is prediction. And this prediction is never easy, because the future, of course, is cloudy. Next one, please. The step actually to do this prediction is, using data mining. And what is that is, in the first step, what we do is where we go to the data that we have at hand, and using, supervised learning or unsupervised learning and data mining, a model is generated that tries to reveal the pattern that is hidden and to understand that behavior. So this model is generated, and then having this model, we are going to use that model to predict future behaviors, okay? So what is important is that one model never is enough. Usually you prepare different models, and keep in mind that all the models are going to be wrong, but few of them are going to be useful. Next one. So linear regression is one of the models that is not, unique for big data. Of course, linear regression is, for hundreds of years around. But it's very simple, but it's beautiful and it's very powerful. And usually when we have a data set and we want to find a model, usually we start with the linear regression because it's very powerful. And a model actually is a representation of the data that capture the most important feature of that data. And when we say the model, usually we're talking about, an equation, a mathematical equation that could be very simple, like in the case of linear regression, the question of the linear line, or it could be very complicated, and that we need this, the supercomputers. And in the linear regression, what we get, of course, is the intercept. We have the slope, which is the coefficient. And basically what we do is we get the model, and we have an unknown. We can plug into the formula, and then we got our prediction, okay? What it does give us this linear regression is the coefficient of determination, the R-square. And the R-square comes from the correlation with V-square, and it's a powerful parameter. Because R-square, let's say, of 90, it means that 90% of the variance of the model can be attributed to the particular variable. For example, if we have the mouse size versus the mouse weight, and that correlation, let's say, is 90.9, that means that 90% of the size can be attributed to the weight of the mouse, for example. So next one, please. So I want just to finish on time. Logistic regression is also very popular, and it's basically used when we have data that are categorical, like a true or false, and via an equation, the logistic equations, we can transform this true or false categorical data into numerical values of probability. So which is the probability of, let's say, that a particular individual is going to be obese, or this mouse is going to be obese. This, of course, you can imagine, is very useful for classification as opposed to prediction. Of course, it can be used also for prediction, and can be used for categorical data and also for numerical data. Next one, please. Decision trees, as I mentioned to you, is very popular. It's very simple, and what it does is try to split the data according to a particular variable. And it's like, when you look at it, it's like an inverted tree, and it will divide the data in order to create subdata as we go down, the tree, that makes these buckets as pure as possible. For example, if we, in this is an example that I will going to play tennis today, and the variables are, your outlook, how the weathercast is. It could be, sunny, overcast, or rainy. You have the humidity, you have the wind, you have, the day of the week, etc. And then you can just, and then the decision is, the outcome is, are we going to play tennis or not? So, and this, according to, to the table that you have, so the decision tree decides that the first decision that divides whether you're going to go or not play tennis is the outlook. You look at, at the weather, and then, the humidity or the wind, etc., etc. Okay, so it's very easy because it's very visual. Even a child can understand how the decision is being made, and it also is very, very simple, and it's very easy to do, and it's very powerful. It becomes even more powerful when you can combine different decision trees, and that is what you have now, a forest. And these random forests are very, very powerful models in artificial intelligence. Next one, please. In this one, we have the support vector machines. The support vector machines are very important to us because what it does is create a discriminator. It creates, an edge in which you can separate two different, let's say, populations. For example, we're going to separate the red from the green balls. So if we have a new ball, according to their weight, for example, most of the red balls are light, most of the green balls are heavy. So if we have one ball that is red, that is heavy, for example, we're going to put it in the green compartment, even though it's red one. So what it does allow us is misclassify without too much penalty. And the example, concrete for us, if, for example, you look at the bottom, is if we wanted to analyze the effect of a particular medication or drug concentration. If you have a lower doses, for example, you're going to have no effect. Then when you increase the dose, there you have the green, then you have an effect, in this case would, cure, the disease. But then if you go a little higher in the dose, you don't have any more cure of the disease. You can have an undesirable effect, which could be toxic. So there is no a particular here age that can separate the good from the bad because you have, overlapping, data. So using this kernel trick, which is basically doing a mathematical transformation, indicate, for example, let's square these values. You have what you see there on the right. So if you square this value, now we can trace a line in this case that could separate very well the undesired red from the desired, which is the green. So the support vector machine are very, very important, useful models. Next one, please. Principal component, I think the last one, is very useful when you have big data in which you have the dimensionality is large. Meaning, for example, we have omics data, you have genomics data, you have 30,000 genes. That's all your columns. Unless you have only maybe 10 animals or 50 people. So the rows are very few compared to the columns. So then it's very, your dimensionality is very large. So it's very hard to see the pattern that is hidden there. So the principal component, what it does is that reduce the dimensionality by just looking at the components that are the most important, the ones that are driving the variability the most. And there, what you have at the bottom, for example, when you have just 3 genes, this is not high dimensionality, you have 3 genes, and then you can see that they are PC1, the principal component 1, capture, most of the dimension, most of the data. And then you have PC2, which is completely independent from PC1, meaning the variables, the attributes that are encapsulated in this principal component 1 are independent that are in the PC2. Okay, that's which is orthogonal. So then you can go to a 3D type of data to a 2D that you can much easily visualize. And you can see this is very important for when you are dealing with big data. So I'm going to stop here. I have more to go, but I think that I'm going just to stop here and maybe entertain a few questions, or maybe I don't know. You wanted to do it at the end?
Yeah, Blanca, if I'll do questions after Jason.
Okay.
All right, so next we're going to have Jason talk about, this is Jason Bates from University of Vermont, and he's going to talk about some applications of AI in healthcare and research.
Can you see that?
Yep.
Okay, so you can see the first slide. All right, so I've entitled this Deep Learning and Its Applications. Let me say that this is a huge field, and I'm certainly not going to be exhaustive by any stretch of the imagination. So what I'd like to do is talk about what deep learning is, kind of where it comes from and how it works in general terms, and then describe briefly an application of it that we are developing in our lab. So this begins really with a discussion of what is an artificial neural network, because that's really the engine behind all these things. If you don't know what it is, you, I'm sure, have heard of it because they've been around for quite some time. So artificial neural networks were actually first started back in the 1940s as models of brain function. Now, this was at the time, actually slightly before the time when computers were available, so people couldn't do simulations, but they started developing the mathematical theory of it, and it's developed ever since, and now it's widely used for pattern recognition and adaptive memory, and it's one of the key techniques in artificial intelligence. And as you're probably also aware, we've had a recent revolution called deep learning, and I'll explain what that is. And this really started around 2012, when the availability of more powerful computers and massive data sets available over the web made it possible to do some rather remarkable things using this deep learning technique. But the actual mathematical methods of deep learning have been known for several decades. Back in the early 90s, when I personally got interested in this field, artificial neural networks were very much of interest. And then they kind of went out of favor and then came back with a vengeance around 10 years ago, and we've been experiencing that ever since. So let's begin by talking about the artificial neuron. And I hope you can see my cursor here, but the artificial neuron is simply a nonlinear summing junction, which is based loosely around what are thought to be the essential features of a real neuron. So if this is the neuron body here, and this is the axon, and these lines here represent the dendrites of a neuron, then a neuron receives input from other neurons. So X0 is an input coming down the axon of another neuron. It gets multiplied by a weighting function, W sub 0, which represents the strength of the synapse connecting those two neurons together. And the same with a bunch of other neurons here, all coming in on the dendrites. And these weighted inputs sum in the somatic body here to be presented essentially to the biological machinery of the neuron, which then decides whether it's going to produce an output or what that output is going to be. So it's a nonlinear summing junction. And critically, the way this neuron decides whether it's going to produce an output or not is governed by a very nonlinear function like this. So below a certain level of summed inputs, which is summed weighted inputs, which is what we're plotting here horizontally, below that, the neuron doesn't do anything. Above a certain level, it fires, and there's some kind of steep ramp up between those two extremes. And this can be a step function, although these days it turns out this has to be differentiable. So it's a smooth sigmoid type function like that, typically. Now, an artificial neural network is simply a collection of these things. You join them together in various ways. So the output going down the axon from one body is going into the dendrites of one of the other neurons and so forth. And there's an enormous number of ways which you can connect such neurons together. And there are many ways in which they are used typically. But the mainstay of all this is what's called the multi-layer perceptron. And that has a structure like this. So we have a set of input data coming in parallel, in each data impinging on a neuronal body up here. And this is actually not a neuron, it's just a staging point, really. And out of each one of these input nodes, you get a weighted contribution, a weighted version of this input going off to all the neurons of the next layer. So this input is weighted by a strength here and put on that neuron. It's weighted by a different strength here and put on that one and so forth. And every one of these input nodes talks to every one of these hidden layer nodes here. This is called the hidden layer. And these are neurons. So these guys here in the middle, they decide on the basis of the summed inputs of what they're getting, whether or not they're going to fire. And when they do, they give weighted contributions to the inputs of all these next layer neurons here, which are called the output layer. And the classic multi-layer perceptron is the output layer gives the classification. So to give a simple example of how this works, for example, first of all, we'll call that the input layer, the hidden layer, and I'll explain why in a second, and the output layer. So multi-layer perceptrons can be trained to recognize different patterns in the input. And here's a very stylized example. And the way that they're trained is each time you present the multi-layer perceptron with an exemplar from a particular class of data, you make minor adjustments to all those synaptic weights to make the output a little bit closer to what you want it to be. And once you've trained the thing enough, you can then give it other examples from the different classes that it hasn't seen before, and it hopefully will recognize them. So if this, for example, is an EKG signal, you'll have to excuse my artistry here, but that's my attempt at making a beat of an EKG. Then you could take every point in that EKG and apply it to a different input neuron, input node. They would then impinge down on the hidden node, and you might have 3 output nodes that determine what classification this EKG goes into. You're either good, you're in trouble, or you're really in trouble. And after training with enough exemplars from all these 3 classes, you've adjusted the weights in these neurons to be able to do that. Now, the hidden layer is essential, and this was a major breakthrough in the theory of neural networks in about the 1980s, I think, when it was discovered that if you didn't have the hidden layer, it couldn't do general classification. But with the hidden layer, it can solve what's called the exclusive OR problem. So if we have 2 input nodes, and you want this thing to be able to determine whether you've got one node positive or the other node positive, but not both, you have to have a hidden layer. And I won't go through this. If you get a chance to see these slides afterwards, you can take the input here as 0 or 1, multiply it by that weight, and then apply it to this neuron, which has a firing threshold of 1.5. So in other words, if the sum of everything coming into this neuron isn't above 1.5, it won't fire. And you can satisfy yourself that if this guy is on, the output node will fire. But if both nodes are on, it will not fire. And if this one is on, it will fire. So that it solves the exclusive OR problem, which is critical for being a general classifier. So in terms of the general classification problem, we can think about this as the multi-layer perceptron defining a boundary in feature space. And I've made a facetious example here. Let's say we want to classify Halloween masks as either being scary or funny. And we do that on the basis of 3 attributes: the amount of hair they have, the length of the teeth in the mask, and the color of the eyes. Now, every point in this space corresponds to a certain amount of hair, a certain length of tooth, and a certain color of eye. So you can represent different masks as points in this space. And let's say the blue points are the funny costumes. Now, where are the scary costumes going to be? Well, they might look like that. They might overlap quite a lot. You might find that there's certain particular combinations that look funny and certain that look scary, but they're not well separated. So what you need to do if you're going to accurately separate those 2 groups is you need a classifier that can define an arbitrarily complex boundary about those points. So everything within the orange line is going to be funny, everything outside is going to be scary. And it's been shown theoretically that multi-layer perceptron, provided you have the hidden layer, can do this. And the hidden layer is essential for this ability. Now, deep networks, because the word deep, of course, is very prominent these days. They do this, they essentially just do this, but they do it more efficiently. So instead of having one hidden layer, they have multiple hidden layers. The hidden layers are typically smaller in the sense of having fewer neurons per layer, but you've just got a bunch of them cascading down. And there's no theory as to say how many you should have to be best, or how big they should be. This is still very much an empirical art. But there's a huge effort out there in computer science departments and Google and so forth to tune these things to be optimal. And then finally, you get the output layer. Now, there's one more wrinkle that we have to add to this to bring us up to the current day, and that is the so-called convolutional neural net. So I've drawn a structure of the whole thing here. So we're going to be moving from left to right. The thing I just described is shown over here on the right, these fully connected multi-layer perceptrons with an input layer, a bunch of hidden layers cascading down to an output layer. And, and here, for example, we may have trained this thing to recognize pictures over here of cats versus dogs, in which case we'd have only two output layers. One would light up if it's a cat and one would light up if it's a dog. And you train these things, including all the weights between these neurons, by exposing it to a gazillion pictures of various cats and various dogs, adjusting the weights each time until finally you've got something that's tuned to just recognize cats or dogs, whatever. But we also have some stuff going on in the front of this, which are the convolutional layers. Now, convolution is simply, in the case of an image, it's simply a spatial filtering. It's cutting out some frequencies in the image and letting through others. And the way the classic convolutional layers work is you have a bunch of filters, and each of those filters is in a square array of weights that are applied to the image and then moved across the image, and the weights are multiplied by the corresponding pixels and all added together. And so for every position of the filter in the image, you get a position over here in the convolve filter. So a little point here goes to that point in the image. And you can do this with a number of different filters in parallel, and then you can even cascade them to have another set of convolutional filters going from the first set to the second, from the first filtered image to the second, and so forth. And at the same time, you can do what's called max pooling, which means you look at collections, local collections of pixels typically, and you pick the biggest one and just retain that and throw out all the others. So the images get smaller here, but hopefully retain the important information. And this compresses the data in the image, but retains the important information. So you start out with an image, you pass it through these convolutional layers and into the multi-layer perceptron. And the final point I need to make is that the filter weights in these convolutional filters are also learned by training in the same way that these synaptic weights are trained. So you end up with a massive number of weights to be trained typically. Now, we've been applying this in the lab, and this is work I've been doing in collaboration with my colleague, Matt Kinsey, who is an interventional pulmonologist and very skilled in machine learning type biostatistics and that type of stuff. We've been applying it to the problem of trying to detect lung cancer by CT. And the problem is as follows. This is a picture of a slice of a lung with a malignant nodule in it. It's a T1 stage, there's the size, it's in the right upper lobe, and you can see there's something there. And that turns out, proven by biopsy, to be cancer. And with the advent now of routine screening of high-risk smokers for lung cancer, hopefully catching it in the early stages, there are a lot of images appearing like this in the medical field. Now here's another image that looks pretty much the same. There's the lump there in the lung. It's in a different place. This happens to be benign. So what's the difference between this and this? Well, just by looking at it, it's pretty hard to say. And even board-certified radiologists have a lot of difficulty doing that. So we are investigating, as are a number of other groups, whether it's possible to train a convolutional deep neural net to recognize whether images like this are malignant or benign. Now, what we would hope is that if we take an image like that and represent it as a single point in pixel space, in other words, every pixel is going off at right angles to all the other ones, it's not easy to draw more than 3 dimensions on, a sheet of paper. Here I've drawn, 5 dimensions or 6? You get the idea. These are supposed to be at right angles, kind of hard to do it. What we would like is for all the cancerous nodules to be green, for example, and all the malignant ones to be over here nicely separated. What we've actually got is something like that. It's a very difficult problem to separate these two groups, but by training multi-layer perceptrons, we hope to be able to do that. One of the challenges is that to train these types of devices effectively, you need a very large number of exemplars of images where you know it's cancer or you know it's benign. This is a fairly small data set that we published a chapter in this book here a couple of years ago. We had 450 cancerous nodules, 845 benign to train the image, and then a smaller number of test images of cancerous and benign. in the hands of, in our hands in this particular case, we got, 75-ish% accuracy at classification, which is not bad. But that still means you have an awful lot of false positives and false negatives. The false positive is actually the big problem, so that people have to undergo nasty biopsies even when they turn out not to have cancer, and you'd like to try and spare them that if you can. So we're still working on this, and we're just about to submit another paper on a bigger study where we've done some development of the methodologies itself. But this type of approach obviously can be used not only in radiology, but it can be used in pathology, looking at different cell smears, recognizing cancerous cells, non-cancerous cells. You could go back to the lung. People have applied it to try and classify patterns of pulmonary fibrosis, for example. You could do it with COPD. One of the challenges is, as I said, getting enough adjudicated data sets to do the training properly. But it's a very exciting field, and I think it's not going to solve all problems, and it's not going to replace all the physicians. So you don't have to worry about losing your jobs if there's any physicians out there. But I think it is going to become an increasingly important aid in at least doing an initial rapid screening of these types of images to speed up the throughput. Just finally, I want to raise something which I find incredibly interesting, which is moving into another area of biology. And you could say, does artificial neural network theory have any relevance for biology itself? And I want to tell you about something that I learned about a number of years ago. This was a science paper in 2003 by a well-known mathematical biologist in England at Oxford called Dennis Bray. And he pointed out that from the standpoint of a living cell, the closest approximation to a neural network is probably found in the pathways of intracellular signals. Why would that be? Well, consider this. Here's my cartoon of a cell. Here's the cell body, here's the nucleus, and here are 3 cell surface receptors. And here are the ligands that join those cell surface receptors and cause the cascade of reactions that happen afterwards. Now, from a formal point of view, these cell surface receptors look a lot like the input layer up here of a multi-layer perceptron. Once a cell surface receptor has been engaged, it starts going through all these well-known second messenger systems which interact with each other and cross over and so forth. This looks very much like a hidden layer. And finally, you get the transcription factors that are produced that go into the nucleus to start producing proteins. This looks very much like the output layer. So different proteins being different outputs. And furthermore, if you think about the way these reactions take place, they're all receptor ligand binding competitive reactions governed by Hill equations, or the Michaelis-Menten equation being the simple form of that. So in other words, they're highly nonlinear saturable equations. So they look like that saturation function that you get in the somatic body. So from a mathematical point of view, this looks very much like a multi-layer perceptron. And if that's the case, a corollary of this is that intracellular signaling molecules are just nodes in a network, which means they're going to take part in a number of disparate cell functions, which explains why you have canonical pathways and non-canonical pathways, and people get very confused about why this should be. But if you think of the cell in this way as like a multi-layer perceptron, it's kind of obvious that it has to be that way. So to summarize, the artificial neuron is essentially a nonlinear summing junction, and it's supposed to represent what is thought to be the key functional attribute of a real neuron. Now, hardcore neuroscientists may become apoplectic when I say that, but to me, I think that really is capturing the essential attributes. But we'll see, that's a controversial point. When they're connected together in the appropriate ways, they gain powerful functionality, and which is related to artificial intelligence, as I've described. The multi-layer perceptron is really the archetypal artificial neural network, and it's used for pattern recognition, and it requires that hidden layer as absolutely essential. And if you increase the number of hidden layers, you get these deep neural nets, which have even increased capabilities. And when you add pre-filtering convolutional layers in the front, the capability increases even further. They all have to be trained, of course, which is the hard part of this. But that's the basic theory of it. And these convolutional deep nets have been proven useful for all sorts of image recognition, including this problem we're working on of distinguishing between malignant and benign lung nodules. But you're probably also aware that some months ago, or was it last year, Google, with great fanfare, demonstrated that they could do incredibly accurate recognition of cats versus dogs, I think it was, by having just training a network on a gazillion images. Once they'd done that, the thing could recognize cats or dogs as well as a human. So it's pretty remarkable and pretty exciting. And finally, I showed you that something that I personally find very compelling, which I think is that cell signaling has formal similarities to the multi-layer perceptron. So this whole idea about artificial neural networks goes beyond just artificial intelligence. It's actually a model of some pretty interesting things in nature generally. And with that, I'll finish. I just acknowledge the few folks in my lab who have been particularly working on artificial neural network with me, particularly Matt Kinsey and some support. Thank you very much.
Great, thanks a lot, Jason. That was wonderful. So thanks to Blanca and to Jason, and to the ATS, to Mei Ling and Liz. So if anyone has any questions, please feel free to submit them in the chat. And if not, maybe I could ask something to Jason first. I was kind of interested in your analogy to the cell signaling network. Although we often think about cell signaling pathways as sort of being on or off, but not like a weighted, a neuronal weight from a synapse. How does that kind of play into it? Is there the strength of a signaling pathway that can be, are there pathways that can be modulated in terms of strength, or are they typically on and off?
Well, they could be on or off, but I would think that the strength of the pathway, probably just off the top of my head, I would think the strength of the pathway is the equivalent of the synaptic strength. And, this, something that struck me a number of years ago when I was listening to seminars around the medical school, as I often do, is that somebody was talking about, I forget in which second messenger system it was, but the canonical pathway was cell proliferation, I think. But this same thing was also involved in apoptosis. And they were kind of sweeping that under the carpet and saying, yeah, whatever, Those are two diametrically opposite functional outcomes. And yet, when you think of the cell in this way, it makes perfect sense that that could happen, because what it means is that the cell is going to respond not to single ligands in the environment, but to patterns of them. That's the point. It's a pattern. It recognizes patterns in its environment. At least this would be the implication of it. So it makes it more complicated to understand, but I think it's a pretty neat insight that this guy had.
Great, thanks. We have a question from David Kasahara. He is wondering, what is the chance for the neural network to make mistakes or biases based on how we teach it? So how would it self-correct? And isn't our input really going to determine the efficacy of the algorithm? So,
absolutely.
Blanca or Jason?
Well, yes. I mean, sorry, I'll just answer. It's entirely dependent on what you train it with. And that is one of the key challenges, is that if you train it with biased data, then, it's going to respond accordingly. But the same is true of our brains. We have biased interpretations of things all the time based on our experience. So I think it's kind of similar. And the only thing you can do against it is keep gathering more data and retraining and getting the thing to have a broader view, I think.
I think also that, as Jason said, the data is crucial. The quality of the data is crucial. The quantity, of course, is important because you need to have a particular, amount of features for the program to learn, the algorithm to learn. And the other thing is that with the neural network, they are very powerful. But I think the drawback, and especially for us scientists, or especially for me, which is, the thing that, itches me, is that it's a real black box. You put the input, you get the output, and they don't tell you what is in between. You don't get the formula, you don't get anything. So, and that, for some people, that could be maybe a little unsettling, because you are going to trust this, but you don't know, the mechanism, what's going on. And actually there are some, I think, some attempts for some data scientists to try to understand what's going on in those hidden, layers that Jason talked about. And I think I read somewhere an article that some scientists could kind of visualize, what's going on in the hidden layers. And you could see, when you have the final figure of your mouse, of your cat, you can see in between how, it's being, constructed. It's a kind of a, like a mental disease, you have all these.
Yeah, that's true.
But
that's true. Yeah, there's a lot of research done on that. However, I would suggest that although we naturally want to know what's going on in the way that we usually think of understanding, I have a strong suspicion that in many cases it's not actually possible to do.
Probably.
And the kind of information that we synthesize in order to make these complex decisions simply can't be written out in algorithmic form. For example, have you ever spoken to a skilled radiologist or pathologist when they are telling you what they see in one of those slides and ask them, really, how do you know that? What do you see? A lot of the time, they can't tell you. And it's not because they don't know what they're doing. They do know what they're doing, but they can't put it into words. They just see the pattern and they know. And I reckon it's the same thing.
I agree.
Thanks a lot to both of our speakers. We've just come up on the hour, so we're gonna stop here. I just wanted to say thanks a lot to Jason and Blanca for your time, and then to keep an eye out for next year's Science and Innovation Center activities at the ATS. And if you could please fill out the questionnaire, the Survey Monkey that's in the chat, for some feedback from our speakers. So thanks a lot, guys. I know there were a couple questions coming in at the end that we didn't get to, but if you want to get in contact with me, perhaps I could get you in touch with the speakers and we could kind of go from there.
Sure. Yeah.
So thank you so much for joining us today, and I hope everyone has a really great afternoon.
Thank you for listening.
Thank you very much. Bye-bye.
Bye, Blanca. Okay.
Bye, Jason. Bye, Mike. Bye, everybody. Thank you.